Hi everyone, my name is Justin Howard. Thanks for taking the time to watch my presentation. Hopefully I can stumble through this enough to get my ideas across clear and concisely. I kind of felt overwhelmed with this paper because the study itself was not too difficult, but just the process of heat shock factor induction is got a lot of moving parts to it. And so I kind of got lost in that for a little bit before I was like, oh, wait a minute, I'm kind of just looking at sigma32 transcription factor. So far, everybody's done a great job. I've learned a whole lot from your papers. I learned a whole lot from this one. And I kind of realized that we're coming to the point where we're using a lot of this stuff we learned about RNA to view biology in a bigger picture, specifically cells and how they function in different environments and some of their mechanisms they have to mitigate stresses. I kind of started thinking about that whenever we were looking at the biofilm paper, and I was thinking about that before I watched Dr. King's latest message, but he kind of said the same thing what I was thinking. I was like, yeah, we're kind of getting to see how all these things work. Some RNAs are blocked by these hairpin structures. Some RNAs, at the point of transcription. Some proteins are regulated at the point of mRNA and their structures. And then what really is different about this paper, I think, is the point of the conformational changes within mRNA that can either turn on or off translation of a protein, not transcription. But this mechanism I'm looking at today specifically is regulated both at transcription and translation, so it's a little bit different for me. But it was super neat to figure out how this sigma32 transcription factor is regulated. So we're going to talk about heat shock proteins for a second in general and the process that kind of goes on behind that, and a little bit of how research led up to the point where my authors, Moriata et al., said we want to understand how this works a little bit better in terms of how this one transcription factor is regulated, or the mechanism behind its synthesis, and how that synthesis is either slowed down or sped up. So the title of my paper was Translational Induction of Heat Shock Factor sigma32, Evidence for a Built-in RNA Thermosensor. It was written by Moriata et al. And published in 1999. This paper was preceded by a lot of research from other people about a decade prior, from what I've gathered. I found one paper as far back as 1983. So let's take a broad view of this process for just a minute. That way I can drill down and talk about the actual transcription factor that they focused on in this paper. So within cells, heat shock proteins are made in times of stress, specifically increased intracellular temperatures. But they're not only just found in bacteria, they're made throughout pretty much all domains of life. Humans, plants, archaean. I'm going to show you an example of those, but specifically one cool thing I learned about them was in humans. Even in biological systems, they can be used as stress markers. I had one ecologist writing about looking at levels of stress proteins in sponges, sea sponges, and spiders. I know a response can be elicited by the presence of heavy toxic metals that are building up, and cells will try to respond to that stressing change. And so if they can take a sample of a spider cell at that point, they can see elevated levels of stress proteins and decide, well, there's a toxic effect happening here. The mechanism is so sensitive and responds so quickly to a change in the environment, it makes it really attractive for these types of uses. Even in humans, the heat shock proteins that are present during, within cancer cells, for instance, here we got red blood cell. This author was looking at the idea that aspirin blocks, or it binds the transcription spot for heat shock proteins on DNA in human beings. That's what aspirin does. And within this certain treatment that they provide here, which is called hypothermia, they just heat the cancerous tissues up to a high level of temperature. I think I read 113 degrees Fahrenheit. If aspirin's blocking the transcription of those HSP proteins, then that means that cell is going to have better survivability. So it may not be a good idea for those patients receiving that treatment to be taking aspirin. And they can be used as markers in other diseases for like inflammation, pancreatic cancer, hardening of your arteries. In Archean, in one of the highlighted organisms in our optional reading, and a few other sources I found, was this Pyrococcus furiosus. I think it means fireball. And so it was it was a pretty big discovery for people when they got down to the hydrothermal fins back in the early 80s in terms of its metabolic processes. Like it can do some crazy things by making its own food and stuff like that. And it likes to live at temperatures of 90 degrees Celsius or greater. One of the other discoveries that came from it was just a better DNA polymerase for better accuracy in PCRs. An author that was in the reading took a look at this and a few other of these type of organisms, these thermophiles, and asked the question, do they have a transient level of heat shock proteins in their system, living at that high level? And then is there a point at which a response is elicited, and how hot does that have to be for them, and what's the mechanisms behind that protein machinery there? Because if they're always in a hot temperature, there has to be something a little bit different happening there. And in their study, they tried to view some other types of bacteria that were in the mesophile type category that like to live in just a more moderate climate, like Escherichia coli. And they didn't find too many analogs to those bacteria. So overall, heat shock proteins, or these chaperone molecules in general, are able to allow cells to sense, and not only just sense but respond to things like exposure to UV light, cold shock. And it was really cool how they did that. They do it by sensing membrane fluidity, and then they can lower the temperature that signals the cell to go into this phase transition. E. coli can elicit a response if it experiences a little bit of change in hydrostatic pressure, changes in pH, oxidative stress, and like I was saying earlier, heavy metals, all can elicit some kind of chaperone response. And that's not even covering the tip of it there. There was a little blip about that Pyrococcus furiosus that was talking about how it lives in extreme hydrostatic pressure. But I wasn't able to connect or spend enough time to figure out if that was related to one of these responses. So heat shock proteins in general serve as molecular chaperones. They modify proteins by folding them, translocating them, maybe across the cell membrane, and they also degrade ones that are not functioning. So you got like proteases and things like that. And all that plays important roles in the cell's day-to-day process and physiology. This is just to name a few things here going on. You can kind of see an example right here. One of the proteins that was focused on in this paper, or mentioned in this paper, was GRPE, chaperone molecule, heat shock protein. So this barrel-looking protein here is GRPE, and those misfolded proteins will actually go down inside of it, and that GRPE will have a change in shape and attempt to fold it, and then when it's done, the cap will come off and release it. So in wrapping up this introduction, let's take a look at all the previous work that had been done before Moriata et al. Asked the question, how is this sigma32 transcription factor synthesis modified or moderated by a secondary mRNA structure at the translational level? Grossman and team noted that sigma32 is unstable with a half-life of 4 minutes. Tilly and some others said that it was probably more around 1 minute, but regardless, the rate of synthesis of sigma32 is correlated with the rate at which heat shock proteins are produced. And Grossman demonstrated this in his 1989 paper. I'm sorry, maybe it was 1987 paper. So he took the RPOH gene that's responsible for encoding sigma32 and fused it to a couple different types of promoters. column A was the lac promoter, column B was the tac promoter, or PTAC, and then C was also PTAC but done at a different temperature. Normally these promoters are repressed, meaning they're not going to transcribe or produce any proteins. But when he added this reagent to them, they were freed up and allowed to synthesize sigma32. So these two black arrows indicate the proteins DNAK and another chaperone protein that was produced by the cells after the promoters were freed up. And so here it's demonstrated that when no sigma32 is being produced, there were no heat shock proteins being produced. But after his prescribed amount of time had passed, he washed those cells, prepared them for gel electrophoresis, and there were dark bands present indicating those proteins were produced. There were no other cellular parts involved in this reaction, so it had to be sigma32 that was preceding their synthesis. It was also noted that sigma32 being synthesized precedes HSPs being synthesized, and that sigma32 was mediated at the translational level. The authors also cited previous work that DNAK, DNAJ, and the GRPE chaperones were observed to negatively regulate sigma32, and these molecular chaperones are present at the cell, or present within the cell at transient levels. But when environmental cues come along, like increased intracellular heat or these other stressors, proteins start to misfold, and then the manufacture of these two molecules ramps up. I also want to make sure that I've gotten the point across that sigma32, we talked about it being an unstable molecule a few minutes ago, but as its levels rise within the cell, it becomes more stable. It's sort of like the clotting cascade or oxytocin in women giving birth. The more oxytocin that's released, the more the uterus contracts, the more oxytocin that's released, and it keeps amplifying like that until the stimulus is removed. To finally round out all the information that Moriata et al. Had in order to make their hypothesis, we're going to look at a couple more studies. The first one being Goff and Goldberg, which they noted that this process of heat shock protein production and this stabilization of sigma32 continue to happen even without an increase in temperature. Bear with me here, I know this screen is getting a little busy. And then second to that is Nagai et al. In 1994 stated that, or was able to document that even if you take this RPOH gene and you modify it to where a certain region on this sigma32 mRNA is silenced, then the heat shock response can be induced normally, but it has no shutdown mechanism. Finally, Strauss came along and said that you can take these DNAK or these chaperone molecules here and mutate them to where they shouldn't be able to function, but when you do that, you can't shut the production off. And so we're going to go to Grossman here for the final cherry on top of this to understand what's happening. So what he did was take a defective lambdoid that had a GalK gene in it that's supposed to encode for galactokinase, that was under control of one of these promoters. He lists PL in his paper, and I've got P1 through 6 on mine, so I'm not sure if they just hadn't discovered it yet, and that's why he called it at the time. Excuse me. But what this is going to do is produce sigma32 and heat shock proteins, and at the same time produce galactokinase. And so what he's going to do is measure the galactokinase and correlate that with the production of these heat shock proteins. And what he found out was in column A here, the strain with the DNA K turned on initially had an uptick in production, but then tapered off after a while. However, in the strain with DNA K turned off, there was a constant rate of production of heat shock proteins. So this graph pretty much shows the same thing. I'm not sure if back in his day they just hadn't named the chaperone molecule GPRE because he calls it F841. But don't worry about the little uptick in GPRE here. What's important is that sigma32 with DNA K switched on ultimately tapers. As opposed to the right side, like we saw in the gel in the previous slide, they both remain pretty much at a constant. And so this decrease in sigma32 on the left side is not related to some decrease in the process of transcription, because galactokinase remained constant. He measured it as a constant rate being produced throughout this whole time. So what he showed was that DNA K controls the production of sigma32, and that the production of sigma32 is moderated at the translational level. Moriata et al. Is able to say, hey, we think that sigma32 is self-regulated, independent from this molecular chaperone circuit here. To begin their experiment, they started out with a gene construct that would contain the bare minimum needed to produce mRNA structures that would ultimately become secondary mRNA structures, and then at that point be able to translate for sigma32 transcription factors. Within this construct, they made base substitutions and then predicted the resulting mRNA structures, foldability, and measured its synthesis at different degrees. You can see here on TLF 247, their control, it's going to be synthesized at 1, or measured its synthesis at a 1 for 30 degrees, and then it had an inductibility of 3. Inductibility means this mRNA has been turned on and is now translating sigma32. So within this gene construct, there are 2 regions that are responsible for this positive and negative feedback loop. Region A is this short underlined sequence here in figure B. There's the start codon. Here is the Shine-Dalgarno complex. And so this region A is masked, or they suspect that it's masked by region B, which is out here, and it's supposed to be highlighted in boldface letters. It's not really visible on this figure here, but you can imagine this is the RNA prior to being folded. They want to predict its foldability and also how much it's expressed before and after heat shock. You can think of foldability as the ability to be conformed to the point where it's starting translation of sigma32. Sort of this armor, you can sort of like this mRNA has been turned on. That turning on would look something like they have here in this second figure, TLF 229, where the start codon and region A is nice and exposed for ribosome binding, and the SD complex is available as well. They've highlighted how 2 base substitutions were made in this particular formation, and it was affecting, and it was made in the B region of this gene. So the B region controls the negative feedback loop, in that it's turned on and conforms this mRNA to where it can be, HSPs can be translated enough to where once there's enough of them made, the sigma32 would become stable again and translation would be halted. Transcription and translation would be halted. That's why sigma32 becomes more stable as it becomes more abundant. And region A is the positive feedback loop here. If it's turned on without anybody there to turn the switch off. It's just going to keep being translated. This table lists all the base changes they made in the gene sequence, and then from that an mRNA was made, and then through enfold they predicted those mRNA's secondary structures. Up here we have the stability of the molecule, with negative numbers meaning it's more stable and positive numbers meaning it's less stable. The expression that was relative to the control, and then its ability, or a score based on its ability to be folded or induced to fold. You can see we've got our control right here with its relative expression measured as 1, or normalized to 1, and at 42 degrees it was 3.5, and then everything else is relative to that. Here are the ones that had a decrease in stability but an increase in expression. And of the other ones that were predicted, these had an increase in stability but a decrease in expression. The expression both before and after is correlated, and he plots this here on this chart. On the left side was 30 degrees. You can see the expression of these mutant RNAs. And on the right side it's at 42 degrees. So they both follow the same trend. However, on the right side at the higher temperature, they're not as tightly packed as they were at 30 degrees, and he didn't have a clear definition of why that was happening. So this is important because it means the expression of the mRNA wasn't dependent on the base sequence substitutions, but on the thermostability of the molecule. The next thing he had to do was make sure these mutant mRNAs were behaving the same within the cell and in vitro. And here he takes a look at these 3 RNAs, the control, 5A, and 181U. And they're significant because they're at the high and low ends of expression for both temperatures. And the results agree in vitro as they did when they were expressed in vivo. You can see 5A was a more stable molecule, and it lasted longer as temperatures increased. And 181, which was less stable, is denatured more quickly. And so this was accomplished with CD spectra, as these molecules are more optically active. They're returning signal here on the left, and then as temperature increases they start to melt or denature, and less signals returned. Up here we have expression in vivo. The solid arrow is the fusion proteins that we're looking at. The hollow arrow is galactosidase, that is like an internal reference, sort of as Grossman did. Whatever's being expressed inside the cell, this galactosidase will coincide with it. So just as predicted, 5A was the less stable molecule. 5A was the more stable molecule. It was expressed less, had lower levels of expression. And then our predicted unstable molecule was expressed in greater levels. So far we have looked at the thermostability and expression of this enfold predicted structure that resulted from 2 nucleotide-based substitutions of GG. Because TLF247 was so markedly different in the enfold prediction, they're going to take a closer look at it and mutant derivatives of changing out some nucleotide bases in it. So here is the predicted mRNA structure from the gene fusion TLF247. And they made a few substitutions in this gene that would alter this mRNA. And these this part of the paper was the hardest to wrap my mind around, so I hope I get this. I hope I can explain this clearly and correctly. So the first mutation they made within the gene produced, they termed it 15A, and it was expected to break this pair, this base pair of C and G here. And then the second one, termed 15A, and 154T were a pair of compensatory mutations. I'm not sure if that's something that resulted. So compensatory mutations, if there's one deletion in one part of the gene, then another gene can alter its nucleotide sequence to pick up for its lacking. And I'm not sure if they induced that or if it's something they observed as they were chemically probing this thing. But it did better, which we'll look at here in just a second in terms of expression and inducibility. And so the third one that's on the low end of expression and inducibility was the 17G122G mRNA structure. So what do I mean by low end? Just like earlier in our other table, 181U and 5A, 17C122G showed both low levels of expression and low levels of inducibility. And you can see this here. Once again, our solid arrow is the fusion proteins, and the hollow arrow is galactosidase that was produced with it. 15A was expressed highly at 30 degrees Celsius, and then after heat induction or heat shift, where the cells were heated up, it was still being expressed. So it wasn't really able to be turned on or inducible. It was just on the whole time. But 15A was pretty quiet. 15A through 124T, I'm sorry, the blue in here, it was pretty quiet at 1.6 at 30 degrees Celsius, and then after heat shift, it became alive here at 7.7 for its inducibility. So what's important about these figures here is that these mRNA structures, 15A through 124T and 15C through 124G, they are compared to this control. Actually, all of them are compared to this control, which is a wild type. This control is just a modified E. coli cells making their mRNA as normal. And the synthesis of proteins from this mutation 15A 124T, that mutation happens to correlate with the complementary sequence of 16S ribosome. So the ribosome 16S is able, its nucleotide bases are complementary to this 15A 124T, and it was expressed more, as opposed to this other one, 15C 124G. It was less complementary to that 16S ribosome, and so it wasn't expressed as much. I'm going to zoom in on this so we can see a little bit better. So once again, he took this control mRNA that was wild type and this 15A 124T mutant and compared the melting curves of them. So once again, at the top here, it's 1, and these proteins are optically active. They're in their primary structure within the cell, and as the temperature is increased, they melt and the signal is lost. And so if you take control and wild type and compare them in this manner, you can see that they are basically superimposed on each other, which really supports the idea that these are thermally regulated mRNAs and not base pair regulated. And I shied away from including the chemical probing in this because really I just could not decipher it. But it's still useful information in confirming that it's a heat-activated mechanism and not a transcription factor which affects the base pairs. Let's take a minute and talk about this toe-print assay. They took a section of this mRNA, specifically sections that started back here at -60 and stretched all the way around to plus 247. Then they added purified 30S ribosomes and this initiator factor tRNA FMAT, which is important because it only allows for the initiation of, or for the binding of ribosomes to the initiation complex, but not elongation. So they're just going to sit right on this ribosome binding complex, but not go anywhere. After reverse transcriptase was allowed to do its thing, it would run around this whole molecule and stop here at 16. And their toe printing analysis, or their toe printing assay showed that. So at 30 degrees Celsius, the whole mRNA was transcribed, and you can see it at the top. It was full-length transcript. But at 42 degrees Celsius, it was stopped at this plus 16 point, showing that it was able to be melted, where that ribosome binding site was open for those purified 30S ribosomes to be united with its mRNA binding sequence. And in figure B, it illustrates how this reaction was effective, and it just sped up at 42 degrees Celsius. And he makes the point that there's nothing else in this solution other than these purified 30S ribosomes and the mRNA that's under scrutiny. So there's no other extracellular markers or cellular components that are allowing this translation to happen. And so he goes one step further to finally put the nail in the coffin that this reaction is thermoregulated, or this site is altered for ribosomal binding related to or because of heat induction. And in figure 6, we see the same thing when it's compared, the wild types compared with the mutant RNAs. So I've brought this SDS page assay here for reference. And if you recall, 15A was overexpressed at both before and after heat induction at 30 and 42 degrees Celsius respectively. And on these two charts, you can see the hollow square in the bottom one and the solid square in the top are both 15A, and it is overexpressed at both temperatures. The other ones we noted follow the same trend. 15A 124T was not expressed, or not appreciable at least at this point, along with 15C 124G and 17C 122G mutants. And so in this final figure, we can see that, again, 15A 124T, which wasn't expressed very much before heat shift, took off again once the temperature rose. And I'm looking at 15A 124T on this bottom section here, B, with the triangle. And it has a definite upshift once it hits 42 degrees. 15A, which was expressed both before and after heat shift, had a showing down here in figure B at 30 degrees, and also was rapidly and generously expressed as the temperature rose. And both those toe prints are appreciated up here in figure A for 15A mutant. It should be noted that lanes 1 through 4 indicate an increase in temperature. So down here at our toe print on 16, you can see that the intensities of the band, like 15A, it's really intense at all times, which we knew it was overexpressed all the time anyways. This is the equivalent of our positive feedback loop wide open without anything to shut it off. Region A is exposed and is wide open. Remember, region A was our positive feedback section and region B was our negative. One thing I forgot to mention was in this toe print essay at the top here, each degree increase step happened at 5 minutes, which I think is kind of important because it shows that not only are these cells able to respond to heat change, they do it rather quickly. And the hotter it gets, the faster it ramps up. So we, or Moriata et al. Has definitely showed that this mechanism of sigma32 transcription factor is not only controlled at the translational level, but is also independent of any other cellular components. And what I think is the neatest thing is that it is regulated by intensity of the heat. And so it can speed up or slow down as needed in order to allow the cell to respond appropriately. So overall, Moriata has done a great job of screening all the factors or ruling out factors of these extra components. I think he was really great at explaining the changes in the base tides, which were related to the secondary mRNA structures, because the structure of this mRNA is critical to its functioning as a switch or as a thermosensor. So it was key to do that. And hopefully I was able to illustrate that for you. I really appreciate everybody sticking with me through this presentation, and also I'm sorry it was late. I had a huge snafu with my computer, but better late than never, I reckon. Everyone, I hope you have a great rest of your week and a great rest of your semester, and thank you for watching and listening to this.